Hello, everybody. My name is Bilal Ali, and I'm a bioinformatics trainee here in the Mayan Lab. Today, I'll be presenting scRNA-seq2Targets, which is a bioinformatics workflow for the analysis of single-cell RNA sequencing data. To begin, I'd like to offer an outline of today's presentation. Firstly, I'll start with some background and discussing some related projects. I'll then move into the implementation of scRNA-seq2Targets. I will then demonstrate its use cases and conclude with future steps. I would like to first introduce some motivation about developing a workflow for single-cell RNA sequencing. Single-cell RNA sequencing is used to measure mRNA gene expression levels in individual cells in a tissue sample. Data is then used to better understand tissue composition and the variability of gene expression across cell populations. As we can see by the picture, the application of single-cell RNA sequencing has been rapidly growing while cost has been decreasing. But single-cell RNA sequencing does pose its disadvantages, as single-cell RNA sequence data can have a lot of missing values since there's not enough mRNA molecules for each gene in each cell. There's many single-cell RNA-seq workflows out there right now, but scRNA-seq2Targets is unique because it focuses on identifying targets and drugs that may be used to manipulate the composition of the profile tissue. I would like to discuss some related projects that inspired the development of scRNA-seq2Targets. The first being the Aptors framework developed by Daniel Clark, a data scientist in the Mayan Lab. Essentially, what the Aptors framework is a way to transform Jupyter notebooks into lightweight web-based applications to allow biologists with little to no coding skills to execute bioinformatics workflows. Specifically, out of the Aptors framework is the scRNA-seq data analysis aptor developed by Minji Jian, a former postdoc in the Mayan Lab. scRNA-seq2Targets largely followed this template of the template of this existing bioinformatics workflow for the analysis of single-cell RNA sequence data. Minji developed the scRNA-seq data analysis aptor that is currently available in the Mayan Lab's aptor catalog. Another project that guided the creation of scRNA-seq2Targets is Multiomics2Targets, which is a computational workflow for identifying pathways and targets for bulk transcriptomics, phosphoproteomics, and proteomics data collected from cohorts of cancer patients. It was led in development by Giacomo Marino, a bioinformatics software engineer here in the Mayan Lab, and was applied to multiomics data collected by CPTAC. Multiomics2Target is an advanced aptor with many additional features that inspired the UI for scRNA-seq2Targets. A paper describing Multiomics2Targets is expected to appear in Cell Reports Method Journal tomorrow. Lastly, some recent analysis applied by members of the Mayan Lab to analyze a single-cell RNA-seq dataset collected from an acute kidney injury mouse model. This unique analysis focused on the difference between proximal tubule cells in the disease state versus the normal state. And for the rest of the presentation, I'll be using single-cell RNA-seq data from this study, which was conducted by the McMahon Lab at the University of Southern California, to illustrate the feature of scRNA-seq2Targets. I would now like to move into the implementation of scRNA-seq2Targets. Here is an overview of the workflow. Users will start by uploading two single-cell RNA-seq conditions, which can be either in plain text or cell ranger format. Then they can select from a variety of pre-processing parameters based on the specifics of their dataset. And then scRNA-seq2Targets identifies and visualizes clusters, performs differential gene expression analysis and cell type identification, uses enricher for pathway identification, uses SIGCOMM links for small molecule reversers and mimickers identification, and finds cell surface proteins that may serve as targets for the cell subpopulation. Here's a look at the data submission interface for scRNA-seq2Targets, which is designed to be user-friendly and intuitive. It provides clear instructions on how to upload data and select parameters for analysis. Users can upload one or two datasets, and when two datasets are inputted, the workflow performs comparative analysis between the two. The landing page also provides the user options for pre-processing and data visualization. Here's a look at the returned output report. After the analysis is complete, results are displayed in a comprehensive report that includes a table of contents for easy navigation, a toggle code button for the users to view the underlying code that's performing the analysis, tables, plots, and other data visualizations. Users can then download the report in various formats using the button at the top. I would now like to demonstrate some of the main features of scRNA-seq2Targets. Firstly, visualization. scRNA-seq2Targets visualizes the data using UMaps, t-SNE plots, and PCA plots. In this example, we can see the differences between the acute kidney injury datasets and how we would like to find treatments that would move the acute kidney injury cells back into that normal condition. Next is clustering and differential gene expression analysis. After the data is visualized and pre-processed, clustering using the Leiden algorithm or user-defined metadata classes occurs. Each cluster or predefined group is analyzed to identify differentially expressed genes in each cluster by comparing it to the rest of the clusters or groups. The identified up and down regulated genes are used for further analysis later in the pipeline. As we can see by the UMAP, cluster 6 was identified as a group of disease state cells. And we can see the top 5 upregulated genes for that cluster in the table on the right. Next, cell type prediction. scRNA-seq2Targets uses the differentially expressed genes to predict cell types for each cluster. And this is done by running enrichment analysis against gene set libraries such as CellMarker, ASCT+B from HubMap, PanGLODB, and others. This step allows us to understand the cell type composition in the sampled tissue. For example, for the previously identified cell cluster 6, we can see that the predicted cell type is proximal tubule cells, which makes sense because they're the most common cell type in the kidney. Next, we move into pathway identification with Enricher. The results from the differential gene expression analysis are then further analyzed using Enricher for pathway identification. Enricher performs enrichment analysis against a variety of pathway databases such as Wikipathways, KEGG, Reactome, and other resources such as Gene Ontology, CHIA, and KIA. This all works to identify pathways and biological processes that are highly active in each cluster of cells. Next, by inputting 2 selected clusters, scRNA-seq2Targets uses SIGCOMM links to identify potential small molecules that may act as reversers or mimickers based on the top 150 up and down regulated genes in each of the clusters. In this example, we can see the potential reversers and mimickers that could be used to push the diseased proximal tubule cells previously identified in cluster 6 back into the normal state. And lastly, scRNA-seq2Targets identifies potential cell surface targets for each cluster by comparing highly expressed genes in that cluster to the expression of those genes in normal tissues from human cell atlases such as ArchS4 and GTEx, and mouse cell atlases such as ArchS4 and Tabula muris. For the highlighted cluster 6, which was previously identified as a cluster of diseased proximal tubule cells, scRNA-seq2Targets identified the following membrane proteins as potential drug targets. Regarding future directions and conclusions, scRNA-seq2Targets offers a large range of analytical capabilities for single-cell RNA sequence data. In this demonstration, we showed how scRNA-seq2Targets offers a unique, easy-to-use workflow for analyzing single-cell RNA sequence data from 2 conditions. We illustrated how data from acute kidney injury and normal mouse cells could be clustered, analyzed for cell type, pathways, reversers and mimickers, and membrane targets. Regarding future steps, we would like to add data imputation, potentially using ArchS4, as will be demonstrated later by fellow trainee Sophie Gideon. We would like to optimize the processes to improve runtime, as well as benchmark analysis accuracy with publicly available single-cell RNA-seq datasets from resources such as HubMap, the NCBI GeoDatabase, and SendNet. I would first like to thank Dr. Mayon and Ms. Jenkins for providing the opportunity to work on this project. I would also like to thank Giacomo Marino, Nashiat Ahmed, and Daniel Clark for their mentorship and assistance throughout the last 10 weeks, as well as the rest of the members of the Mayon Lab and Bioinformatics trainees.